Type 2 diabetes, outdating junk DNA. Morgan Escobedo, BI308, spring 2025. The purpose of this project is that I selected a phenotype of interest, type 2 diabetes, and identified a gene of interest from a GWAS database, TCF7L2. I wanted to identify if my gene is essential or not by applying an evolutionary approach and identifying if my gene needs further study. I hypothesized my gene is not causally related to type 2 diabetes. My gene isn't essential and has the same rate of evolution as the expected molecular clock model. I chose this phenotype because my dad is Hispanic and has type 2 diabetes. And I found my phenotype of interest through a gene database. And there was no changes in settings. I just did some playing around and found this most interesting. So diabetes mellitus type 2 is a long-term condition where the pancreas doesn't make enough insulin. The disease causes cells to respond poorly to insulin and take in less sugar. Insulin lets sugar into the cell to fuel muscles and other tissues. I chose specifically the population South Asian, East Asian, European, and Hispanic. My SMP results from the GWAS protocol. I found this through gene database, and then once I selected my phenotype of interest, I found my gene of interest. And there was no change in settings as well. So I identified my SMP, and then I found that it was an intron, which is a non-coding region that affects splicing and gene expression. So it changes expression, but not the function. So the population that I listed in my phenotype of interest shows that the alternate allele frequency is 28.7%. The T allele of the SMP of my SMP in the gene has that frequency, and this shows that it is a common disease, common variant, and the common disease, common variant hypothesis would back that the common disease that I chose, type 2 diabetes, are influenced by many common variants, specifically the T allele, that contributes to an increase in the risk. This aligns with the idea of the low percentage. So that because a person carries the T allele doesn't guarantee they'll get type 2 diabetes. The prevalence of type 2 diabetes in the population remains lower, which supports the CDCV model, rather than suggesting a strong deterministic genetic cause. And then ClinVar showed that the SMP doesn't cause disease directly, but increases susceptibility, and that the T allele have higher gene expression in the pancreas or liver. And the functional information makes a strong case that my SMP is not just statistically associated, but likely functionally causable by the population. So then this is my gene transcription factor 7, like 2. I found this through the gene database and through the GWAS. And to describe it, there's 20 exons, 19 introns. I found the location, the length, and how well it is studied. And I found this through PubMed, and I made a chart. And to summarize, it was studied 2,054 times starting in 1990. The volume of data reflects a high confidential level in the biological relevance of the gene. It's been extremely studied, and in my opinion, doesn't need further, and is not further research is not necessary. So then, the knockout, not there yet, but the protein that I found this through Wikipedia. I searched up my gene and scrolled down to proteins and selected the first one and found the query length, which I will use in a couple slides. So the knockout, I did this protocol through mousephenotype.org and entered my gene of interest, and the following came up that this significantly was impacted by the knockout. So the adipose tissue was involved in fat storage and metabolism. The distribution leads to leaner body composition, indicating regulation of insulin signaling, and this aligns with the known role of type 2 diabetes. especially for my SMP, which is often linked to insulin and glucose regulation. Then growth in size body region, the knockout enhances muscles or non-fat tissue mass, suggesting that my gene may inhibit lean tissue development under normal conditions, again tying to the energy regulation and metabolism in type 2 diabetes. Then the mortality in aging, complete knockout causes death before weaning in all causes, proving that my gene is essential for survival, likely due to developmental issues or failure in critical regulatory systems. This strongly supports its classification as an essential gene, which goes against my hypothesis. So these effects span multiple systems, confirming that it is just not correlated with phenotypes like type 2 diabetes, but is causally involved through broad transcriptional regulation. Again, countering my hypothesis. So now, my alignment results. I found this through Wikipedia. Blast found all the additional putative ortholog sequences of an additional 13 species, and I chose this that all match closest to my query length, which was 602. Then I imported it into Eugene, selected cluster O and then all default settings. So what came out was few variable regions between species, which means it is more conserved among each species. This interprets functional importance. Conserved sequences tend to be biologically important. Evolutionary has preserved them over millions of years. And this supports the idea that junk DNA isn't always junk, which was why my title was based off of it. Next is the gene tree. I used Eugene for this, put in the setting the file ml tree, and then the default settings, but selected open in a new window. So I chose, and then I used text editor to create a new file for it. And I used the maximum likelihood tree because it is the best mix of accuracy and reliability without being too slow or too simple. It's more accurate because it uses real models of DNA or protein changes over time. So this is it compared to the consensus tree that we were given in class. Tree reconciliation. The distance between the trees of the two workbenches is 0. And I used this because the molecular clock is a null hypothesis, and it comes with assumptions, and these assumptions can be tested through the tree reconciliation. And because the distance is 0, it means the assumption is met, and we can have more confidence in the molecular clock hypothesis. So my molecular clock shows that, as you can see, it's scattered and does not go along with the slope line that goes through the origin. And I got this through calculating linear regression of genetic distance by divergent times. I used the pairwise genetic distance among 14 taxons that was given to us in class. The alligator was chosen as my outgroup, and I used the log MYA because it shows a different scale by taking scattered data and scrunching it down. And this cluster, the upper cluster, I interpreted as the longer branches, which showed that there's more genetic drift, and the lower branches, where the shorter branches showed fewer differences. And the clusters mean that the gene did not change at a constant rate over time, also suggesting the gene is under purifying selection, where mutations are removed over time to conserve the sequences involved in the biological process. Next, I compared it with others, and my slope is moderate, which shows that it is faster evolving than the housekeeping genes, but more conserved than many other genes in the class. It indicates my gene likely plays a big role in certain pathways, and the moderate R2 gene shows the R² gene doesn't follow the clock under selection pressure, just as the graph showed. And my conclusion is my gene is not essential. My gene is essential. My apologies. My gene is causally related to type 2 diabetes based off of the rate of evolution, the R² value, and the knockouts. All support that it is a causal relationship between my gene and type 2 diabetes as well as my SMP. And I do not recommend further study based off of how much it has been studied already. So even though that my SMP is an intron, it is not just statistically associated with the disease, but it is causally impacted. And I found this very interesting because my dad has type 2 diabetes, and that I didn't realize that introns could have such an impact on a disease.